things we're doing this year is tissue testing. And we're sending lab samples away and doing some tissue tracking. And so we are sampling every Monday of the week. Last week was our first sampling. We collected soil samples from roughly a 5 by 5, so 25-foot square. We're also collecting leaf samples of the first fully developed trifoliate there. And based on that, we want to see how our crop is doing. And we're also going to conduct some foliar fertilizer applications and see how that responds as well. So here's our soil test report from our samples collected on June the 24th. We have a few concerns that kind of jump out here when we're looking over this. So first of all, we're in a loam-based soil. You can see based on our cation exchange capacity, not too heavy, but fairly high pH, as well as we're definitely lacking in our potassium fertility there as well. As you can see, especially in the disc grip and the 2680H treatments, we are pretty low there. Definitely lacking a little bit in our organic matter as well, especially where the disc gripper was applied. Don't know if there's a correlation there with tillage type. We have been doing this tillage now for 5 years consistently, and so another thing that jumps out at us here is our potassium to magnesium ratio, definitely out of whack due to those low potash levels. So something we're going to have to look at in investing in some fertility and trying to build some soil tests. Now let's spend some time and look a little bit more closely at some of our tissue tracking from 2020. So this is one of the tissue tracking reports we have from Honeyland Ag Services, where we're right in this instance looking at the 2730 disc gripper. But you can see there's an individual chart for each of the main nutrients that we were tracking throughout the year. You can see all the different sample points represented by dots. And then there are also different colored lines, where our black line is our sample that we collected. The orange is the average of our samples. And then the high and low, high is in green, low is in red, of the samples collected by Honeyland Ag across a similar growing area. So interpreting this information is a little bit complex, but we're going to try and break it down nutrient by nutrient here. So first off, let's focus on nitrogen. Let's take a look first at the graph on the left-hand side. This is research published by the University of Illinois, where they looked at crop utilization of various nutrients, in this case nitrogen. There's different colors on the charts representing the usage in percent by the plant for different nutrients in different parts of the plant. For example, in this case, let's focus on the leaves, since that is what we were tissue sampling. So let's look at the green part. So you can see it ramps up around that V4 stage, peaks around R5, and then that nitrogen gets translocated into the grain around that R5 stage up until maturity. So on the right-hand side are our own samples from the 2020 plot in Exeter. Let's focus on the black lines in each of these graphs. Each graph represents one of the tillage treatments. You can also see a green vertical line on the graphs, which represents when our foliar fertilizer was applied, which did include 12% nitrogen. So we can see similarly around that V3, V5, we have a ramp up in increase of nitrogen into the leaf. Around that R5 stage in the middle of August, we see the nitrogen in the leaves start to fall down. And this is likely because that nitrogen is being translocated into the actual grain. So we definitely seem to follow the same trends as the research shows. So now looking at phosphorus in the plant, let's first focus on the left-hand side, where we're looking at the graph from the University of Illinois research. When focusing on the green area, which is the nutrient in the leaves, we see an uptick around V4 again, and then a peak around R5 and a decrease where the phosphorus is translocated into the grain later in the season. Now looking at our graphs on the right-hand side from our actual samples and the different tillage treatments, this time we actually have two foliar fertilizer treatments that were applied. So we have the green as well as the yellow, which was phosphorus and potassium foliar. And interestingly, after that second application of phosphorus and potassium, we can kind of see a bit of an uptick in the nutrient content in the leaves. But then following the research around that R5 stage, we see the phosphorus in the leaves drop down as it's being translocated into the grain to make the soybean. So generally speaking, we follow the same trends, but we did see a bit of an uptick in nutrient content in our leaves due to that foliar fertilizer application. So now let's look at potassium. So first off, again, the graph on the left-hand side from the University of Illinois. This time in green, the leaf nutrient content rises much more gradually to R5 and falls down a lot more gradually, indicating a lower percentage total of potassium in the leaves, whereas the majority is within the stems and petioles, giving it strength. Now, on the right-hand side, when we're looking at our tissue samples from 2020, we see kind of a more gentle slope, definitely, to our samples, but also, interestingly, looking at the yellow vertical line and the sample following that with our 01620 foliar fertilizer application, we do see an increase in potassium in the leaf tissue due to that foliar fertilizer application. However, it does seem to quickly peter off and fall down as the soybeans get more mature. Now let's look at sulfur uptake. So looking at the University of Illinois graph on the left, the trends are very similar to how nitrogen is used within the plant. So we see a quick increase around that V4, V5, up to a peak around R5, and then a gradual decline up till maturity in the utilization of sulfur. So looking at our samples on the right-hand side, very flat. We don't see a lot of trend there in terms of sulfur content in our leaf tissue. Now, there is a slight uptick of sulfur after our foliar fertilizer application around July the 1st, I believe, was the application date. However, nothing too significant. Last, let's look at manganese. And so we can see on the left-hand side again, the University of Illinois research shows an increase of manganese pretty significantly after V7, and then it plateaus until close to maturity where there is a slight increase. However, there is not that decrease that we've seen with other nutrients. Now, what's really interesting is in our samples, we see this exact same trend occurring. Now, another interesting point is our foliar fertilizer applied on July the 1st, the green vertical line on this graph, was a manganese-based product. However, we did not really see anything in the way of leaf tissue test to show us that there was a strong uptake of manganese into that plant. Now, all of this being said, even though we did see evidence of a nutrient uptake within the tissue samples, this did not translate into any yield differences in our soybeans. We had no statistically significant yield differences between a foliar fertilizer of manganese and the potassium together, or the manganese alone versus none at all. There are many more demonstration plot videos. Head to our website, hurontractor.com, select Precision Ag, Demo Plots, then select your plot of choice, and choose from a list of YouTube videos from throughout the year, as well as other harvest plot results